proud to be a second-year recipient of Cure Cancer funding. And just to tell you a little bit about one of the projects that I'm working on. So the disease that I focus on is, if it clicks through, there we go, is lung cancer. And this is quite an insidious disease because patients tend to present with these late, large, advanced tumours, such as what we see in this radiograph here. And this is somewhat what contributes towards the disease being the leading cause for cancer-related deaths worldwide. But also associated with that, it has quite a poor, comparatively poor 5-year survival rate of 17% here in Australia. So a lot of what drives my work is finding better ways of diagnosing and treating these our lung cancer patients. In speaking of treatment, we heard this morning about immunotherapy, and it has indeed been a game changer for lung cancer, but unfortunately this doesn't benefit the majority of patients. So unfortunately, we're still reliant on chemotherapy, which forms the cornerstone for a lot of the treatment for the majority of patients. But we're looking at older data here in the table on the left, but the same issues still exist, that really only 30 to 40% of patients are surviving the first 12 months. And a lot of this is because all patients develop a resistance to their therapy. Now, how this therapy works, as you can see in this lower cartoon, is by attacking the genome of the tumour cell, by creating this irreparable DNA damage, which then forces the tumour cell to go and die. So for this reason, I've been looking at trying to identify novel genes or molecules that are involved in the repair of these DNA damages that we might be able to exploit to delay or prevent therapy resistance moving forward. So for this reason, I've been studying the molecule cell division cycle-associated protein 3, or CDCA3 for short, and less of a mouthful. And what I've identified is that this particular molecule is markedly overexpressed in lung cancers versus non-malignant tissues. But also we have this spectrum of expression through high through to low expression for the molecule. And particularly pertinent to improving chemotherapy response, we found that levels of CDCA3 correlate extraordinarily strongly with how much DNA damage exists within a lung tumour. What we've also found is that the levels of CDCA3 are very strongly prognostic. So we look at the curves, we can look at patient survival over the course of time. And we see the patients that have higher levels of CDCA3 within the tumours have a drastically poorer survival outcome than those patients which have lower levels of CDCA3 in the blue line. So I've really been focusing on how to improve these outcomes for lung cancer patients, but also focusing in on these CDCA3 high patients. So what I've done is I've spent a long time trying to work out what's the actual function for this molecule within the tumours. And what I've identified is that this protein or molecule, CDCA3, physically interacts with this other molecule, which I've just termed enzyme K. And this physical interaction promotes the activity of enzyme K within our tumours. And this enzyme K is a master regulator for control of cell division. This interaction then promotes cell division, which leads to tumour proliferation and tumour growth. But there's also a number of chemical drugs which target this enzyme K or inhibit its activity. And one of which that I'm particularly excited about, but I'll just refer to today as drug X. And we found that drug X performs a lot better in tumours with higher levels of CDCA3 than in those tumours with low levels. So the lower the number, the more potent the drug. So with the help of Cure Cancer, we're investigating this therapeutic opportunity that we have. So in these CDCA3 high tumours, combining drug X with chemotherapy or with targeted therapies to block this cell division, globally speaking, to then induce tumour killing, an anti-tumour effect. So what we propose is this diagnostic strategy. We can stratify patients based on CDCA3 levels to help guide patient treatment moving forward to improve the response rates to chemotherapy. And with that in mind, we've developed a robust technique to detect CDCA3 levels in tumours, as shown here in the brown stain, from low through to high. And this approach is now being patented here in Australia. And more excitingly, we're also now in high-level talks with the manufacturers of drug X to gain exclusive license to this drug in lung cancer indications. And why this is quite exciting is because drug X is a late-stage clinical asset, and it's going through phase 3. It has a well-characterized safety toxicology profile, and that really does enable us to move more quickly into clinical trials to improve the outcomes for lung cancer patients. So I'll just leave it there, but I just want to pay tribute and thank you to the following people: but also my funding partners. But in particular, I'd like to thank the Denton Family Trust and Cure Cancer, really to enable me to help grow my little team and to create some impact. So thank you very much.